So good afternoon, friends. Today, our topic of discussion in channel is radiation-induced DNA damage and its consequences. Radiation-induced DNA damage is a requirement in the radiotherapy for killing the tumor cells, but some of the naturally induced radiation damage by different methods like viruses, sometimes by or different causes. These radiation-induced damages lead to the some disastrous consequences, and as well as the radiation itself causes the sometimes. So we'll be exploring in this presentation all the consequences which happens. So actually DNA is the principal target for biological effect of radiation-induced cell killing, mutation, and as well as carcinogenesis. When ionizing radiation pass through the tissue, result in damage to the DNA, mainly through the effect of free radicals. So you see here in the figure, there are two kind of DNA damage by radiation. One is the direct DNA damage, and second is the indirect damage. The indirect damage happens with the photon when you fall in the atom, and then since the cells are plenty of water, more than 70%, that will hydrolyze the water, and then free radicals damage the DNA, and that is called indirect damage. And direct damage, which directly a photon hit the atom and the discharge electron damage the DNA segment. So these are the methods in which the indirect having a predominant phenomena in radiation. Almost 75% DNA damage happens with the help of free radicals, and direct damage is limited up to 25% cases. So damage to DNA result in immediate cell death, one consequences, or in the next mitosis, or as a result in a permanent change in the genotypes, means the mutation which transmitted in the future generation cells. So low dose radiation is more likely to cause changes in the genotype, because the high dose radiation will kill the cells and end the debate of radiation damage, because the cells itself got killed. But in the low dose radiation is cause a damage to the genotype after DNA damage, and that transmit the future generations. So what actually the DNA damage consequences are? Like there are few genes that is normally present in the human tissues, and these genes are very helpful, like the p53 genes mapped on the chromosome 17. These p53 gene, when they are present in the normally, they help in the cell cycle regulation, and whatever the minor damage happened of a DNA, it repaired itself by the help of p53 gene. And if they are not able to repair the DNA, they move the cells towards the systemic death, that is called programmed cell death, that is also called apoptosis. So when the cellular damage occurs, p53 arrests the cell cycle until the damage is repaired. If damage at all cannot be repaired, the apoptosis happens, so there is no problem happens in the future generation or in the same generation damage. But if a segment of a DNA got damaged, and then that contains the p53 gene, so that mutated p53 gene becomes more harmful, because the DNA damage is not getting repaired because of p53 gene, which is likely to repair this thing, is get itself mutated. Cell cycle will continue without the help of p53 gene repair, and then the cells becomes cancerous. So mutated p53 gene does not arrest the cells to the cell cycle, and damage will continue and divide, and that will result in a cancer. There are different type of cancers with the genes play a very vital role. That is called tumor suppressor gene, like also called oncogene, leukemia, breast, colon cancer, gastric cancer, lung cancer. There are CMYC amplification of these oncogene. This lead to the cancer. In case of renal cell carcinoma, one nipple dysfunction. These gene dysfunction lead to the development of a renal cell carcinoma. CML, we all know ABL, BCR, proto-oncogene translocation lead to the CML. That I will explain you in the future slides, further slides. The childhood neuroblastoma or a small cell cancer that lead to the because of amplification of NCMYC gene. So the C is responsible for leukemia, breast, colon, gastric cancer, or lung cancer, but NCMYC gene amplification lead to the childhood neuroblastoma and small cell lung cancer. Follicular lymphoma, BCL2 amplification, CMYC mutation. BCL2 means B cell lymphoma virus, 2 amplification. Sporadic thyroid cancer, RET mutations, and familial melanoma, P63 mutation, familial breast and ovarian cancer, BRCA1, BRCA2, invasive ductal carcinoma, breast, HER2 amplification, colorectal cancer, and gastric cancer, APC gene mutation. So these are the actually gene responsible with a different kind of a cancer. We should know all these things. So DNA is a large molecule with a well-known double helical structure, which all we know. It consists of two strands which held together by the hydrogen bond between the bases. Each strand consists of alternating sugar and phosphate group, and the sugar is basically in DNA is deoxyribose attached to the 4 bases here. Sequences is, and because of its sequences specify the genetic code. Like 2 bases are single ring group, like pyrimidine is a single ring group and having a thymine and cytosine nucleoside, and 2 bases are double ring group, that is purine, that is adenine and guanine, that we all have read there in the plus 10 plus 2 classes even, and a base on opposite strand are complementary to each other, like A pair with a T, adenosine pair with a thyrosine, and guanine pair with a cytosine with the help of double and triple covalent bond respectively. So you see here how the combination of in DNA strand bond is there between G and C, A and T, and you see here the single ring compound like cytosine and thymine, they are purine bases, and the double ring compound that is purine, that is adenine and guanine. You see the double ring here. So an X-ray and a gamma rays break off a single strand occurs. If a single strand is break by the radiation, it repairs automatically because the opposite strand work as a complementary strand as a template for repair. If repair is incorrect, mispair, then mutation occur. But in so the single strand break of a DNA by radiation is not a problem because the opposite strand act as a template and repair occurs and nothing amiss is happen usually. But if both strands of a DNA are broken and breaks are well separated, the repair again occur readily because two breaks handled separately. How it happens, I will show you in this figure. Here you see two strands are broken and breaks are well separated to each other. So like E and A segment is got broken, then AD segment in the lower figure, AD segment act as a template and repair the opposite segment. And suppose in the lower segment of a DNA strand, CT segment is broken, then GA act as a template and the repair occurs. Double-stranded break happens, but break is separately, then it is repaired automatically. And for single strand break, the upper figure shows that the one is strand is break a portion, the opposite strand back act as a this thing as a template, so there is no problem happens in the future generation. And the similar things are written here, how this happens, which I have already explained. The double-stranded break repaired by two processes. There is one is homologous recombination, that is favorable, and illegitimate recombination, that is unfavorable process. So B is dangerous. Homologous recombination is favorable, nothing, no problem happens. On radiation double-stranded break, the single-stranded break ratio is usually happens by radiation. So the single-stranded and double-stranded ratio is between 1:25. Means single-stranded break happens more readily than double-stranded break, double-strand break in DNA. So you see here, the homologous recombination is an error-free process. You see here the break is happening in the top figure, but the portion of opposite DNA segment, the chromatid, they repair as act as a template and repair happens. And then the similar happens in the upper segment of act as a template and repair happens to the other chromatid. Non-homologous. Now, our break happens opposite to each other. Direct break, end-to-end joining of breaks happens, and then some fragment is being chipped off from here, then a mutation happens and then lead to disastrous consequences. So that is why it is called non-homologous recombination. Illegitimate recombination is a dangerous thing, unfavorable process. You see here what happens here, the two non-homologous chromosomes exchanging a part of a long chromosome. You see here, that is called reciprocal translocation. One chromatid portion goes to the other chromatid chromosome, and another chromosome's portion got replaced with that portion. So this is phenomena is happened in the chronic myeloid leukemia. I will further explain you how it happens. ABL BCR gene. You see here, that is the gene fusion was found like in CML, in which the ABL oncogene fuses with the BCR gene as a result of a symmetrical translocation between the chromosome 9 and 22. You see here the normal chromosome 9 having ABL gene and normal chromosome 22 having a BCR gene. If these are present, they are normally present in all human tissues. But in the chromosomal translocation, what happens? The translocation happened between the 9 and 12. The ABL and BR is combined together and found in the one chromosome, that is called Philadelphia chromosome, and that is called BCR ABL breakpoint cluster region and ABL gene fusion, and that lead to the chronic myeloid leukemia. A similar picture happens in the some sarcomas as well. So here the DNA damage strand help in the process of damaged DNA strand repair. A non-homologous recombination, illegitimate recombination is an error-prone event, probably account for many of the pre-mutagenic lesions induced in the DNA of human cells by origin radiation that I have already explained to you. So one thing is also very important, that the radiation which is transmitted in the human body now how it energy deposition happens. The energy is not deposited uniformly in absorbing medium, but it is located along the track of the charged particle. In case X-ray, gamma rays, or in case neutron, proton, alpha particle, radiation energy defined in terms of spurs, blob, and short track. What is spur? Spur contains up to 100 electron volt of energy and contain an average 3 ion pair and remains limited in 4 nanometer diameter, particularly in the gamma rays and X-rays. 90% energy deposition occur in form of event as a spurs. So that whatever the clinical practice we are practicing X-rays and gamma rays, like X-rays in the linear accelerator, gamma rays in the cobalt, the energy deposition occurs in form of spurs, 95% energy. And that energy is limited in that 4 nanometer diameter. This is 4 nanometer diameter is important, and it produces a maximum DNA damage, because you see the DNA diameter that is 2 nanometer. So 4 nanometer means it overlaps the DNA and does the multiple site DNA damage. There is another form of energy deposition that is in form of blob that remains it's a wide area but 7 nanometer diameter, and it contains 12 ion pair, almost 4 times higher than the spur, and diameter is also 2 times higher than the spur. So that the blobs are basically less frequent with the gamma rays and X-rays, and blobs are usually produced in case of densely ionizing radiation. So whatever the X-rays and gamma rays, we know that it's a partially ionizing radiation, but densely ionizing radiation produce blobs and are like densely ionizing radiation. We all know that it is produced by the alpha particle or neutrons. Thus, more damage is produced, and such damage could be difficult to repair. Both single and double-stranded DNA break can measure readily by isolating DNA. So, what is the process by which we see that how much DNA damage happens? There are electrical methods by which we found it. What are these methods? For both single or double-stranded break can be measured readily by isolating DNA from irradiated cells. You first irradiated the cells, and now that causing the pieces to pass through the porous or such as a gel filter, the broken DNA segment through the filter or under the electrical field in the gel, using the fact that the DNA is a positive charge, so that it will go towards the negative. So smaller and broken pieces of a DNA will move towards there to the to negative to the cathode area, and from where it can be separated and measured. Then how much DNA damage happens. So DNA in the cells are more resistant to radiation than the free DNA in a study. Why? Because the free cells do have a free radical scavenger. Due to this presence in the cells, free radical scavenger mop up the free radical produced by the radiation, which is causing to damage the DNA. So that's why the free scavengers help in the damaging of the DNA. Radiation induces large number of lesions in DNA, most of which are repaired successfully by this process. So let us define what is the D0 dose. A dose of a radiation that induces an average of one lethal event per cells leave 37% still viable cells. That is called D0 dose. The D0 dose is defined in a mammalian is usually between the 2 to 3 gray actually. As a slide showing the 1 to 2 gray is basically 2 to 3 gray. So that if 2 to 3 gray dose is given to that particular tumor cells, numbered tumor cells, They will kill all the cells except leaving the 37% cell still viable. Means 63 cells will kill and 37% cells will be still alive. That is the concept of dose fractionations to kill the subsequent cells in the further fractions. So, on the D0 dose, and practically we have detected that after the D0 dose, what damage happens? Base damage more than 1000, single stranded break is 1000, double stranded break almost 40. So these are the data. Single strand break does not lead to the all killing, but the cells killing better correlated with double stranded break, and double strand break are more relevant lesion lead to most biological insult. Example, double-stranded break lead to the mutation, radiation carcinogenesis, and chromosomal aberrations. Now the chromosomes and the cell division, the largest part of life of any somatic cell is spent in interphase. During this, the nucleus of the cells appears as a fine lacework of a lightly stained material in interphase. Nuclei are also seen in various events that occur during the mitosis. Each chromosome has a lightly strand constriction known as a centromere, and a prophase phase of a cell cycle end with the disappearance of a nuclear membrane and lead to the stage of a metaphase. Prophase, then metaphase, and then telophase. In metaphase, the chromosomes comes at the equator, spindle formation happens, and in the telophase it ends with the mitosis. In this phase, the nuclear membrane at a pose of two nuclei form. So the cells time of the end of a mitosis, and then the two daughter cells is being formed. The telomere basically cap and protect the terminal end of a chromosome. Each time after the normal cell division, somatic cell division, there are shortening of telomeric part of DNA. After each cell division, it gets shortened so subsequently, and after 40 to 60 such divisions, telomere in a chromosome shortened dramatically, and then no further help from a telomere the cells can acquire. You see, in a young age, the telomere portion is quite longer. In the elderly age, the telomere portion is quite shorter. And telomere is very having an important. Telomere have a set number of nuclei, GG, ATT. So this telomere, after the repeated mitosis, since DNA significant loss, at this point cell cannot divide further. Thus the telomere has been described as a molecular clock. But in case of stem cells and cancer cells, this telomere does not help. Why? Because this problem is by activating the enzyme telomerase, which rapidly synthesizes the telomere shortening after each cell division. So telomere length is maintained. The cells become immortal, and this is happened in the cancer cells. And 90% of human cancer cells of the biopsy specimen exhibit the high telomerase activity. In the radiation-induced break, incorrect joining of damaged chromosomes may lead to chromosomal aberration. Each of effect of ionizing radiation are described in terms of their appearance when the preparation is made in the first metaphase after exposure to the radiation. Metaphase is a phase where you can assess the radiation, ionizing radiation effect. Breaking of DNA after radiation, the broken end are usually sticky and rejoined with the other sticky end, a broken end joined with the normal or unbroken DNA segment, both. Once the DNA break is produced, what are the possibilities? Broken end joined together in original configuration. If they join original configuration, no problem, no nothing amiss is visible at a neck meiosis. But the break may fail to rejoin and give rise to aberration, deletion seen in the neck mitosis, that is dangerous. Broken end may reassort, rejoin, and broken end to give rise to the chromosome that appears grossly distorted in neck mitosis. So, what are the radiation-induced chromosomal aberration? One is ring type, second is a dicentric type, third is anaphase bridge. All three types of aberration caused by gross chromosomal aberration, and that becomes clearly visible. So that you see the ring type and the DNA segment is damaged, acentric segment lost, and then a portion of two sticky ends joined together and a ring formation happened. And after the second mitosis, they overlapping the second ring formation, and then that happens. In dicentric type, when a break is produced after irradiation of a cells, here when the break occurs in two separate chromosomes, they are sticky and joined with each other to form dicentric aberration, and acentric fragment will be lost, which is shown in the figure. In a phase bridge type, chromosome aberration, breaks occur in each chromatid of a same chromosome, incorrect joining of sticky end of formation of sister union in next anaphase. But when the cell's division occurs, joined end of chromosome during the anaphase will be stretched out due to opposite pole. The separation of a daughter cell is not possible. This is a lethal type of chromosomal aberration. I'll show you in the figure. You see here, break happened to both the chromosomes, and then both chromosomes attached to the centromere are rejoined, and after the second division, they will stretch out, and then the problems happens, the lethal aberration. Two non-lethal type of chromosomal aberration is symmetrical translocation. Here the one portion of a segment of a DNA goes to the other segment of a DNA, and this other segment of a DNA is coming to the other segment of a DNA. This means that translocation is usually found in the several human nuclear malignancies like PNAT tumors. And the second is symmetrical translocation. This is called symmetrical translocation. The chromosomal aberration in the human lymphocytes, you can observe in the peripheral lymphocyte, have been used commonly as a biomarker of radiation exposure. That I have already explained in the earlier chapter, that for radiation-induced damage, the lymphocyte is the best, this peripheral lymphocyte, to assess the amount of radiation and the radiation damage. Blood sample obtained after the few hours of total body irradiation. Frequency of chromosome aberration reflect the dose received by the DNA. So that's the topic. Thank you very much.